myself.
Okay, so seems we are live now. So I'm starting with the workshop. So welcome everybody. So this is a 45-minute workshop where I want to give you an introduction how we can use the Spectra package to analyze mass spectrometry data in R. So what you see here, maybe I post this also in the chat. You can get the workshop along with all the data and everything from this page here. And just a very short information on the workshop itself. So what we are going to do here is essentially these 4 steps. So first of all, we are importing mass spectrometry data from MSnbase files. So this is the, let's say, standard file format to store mass spectrometry data in. Then we will select certain MS2 spectra for a specific compound, and we will then compare and match these spectra against a reference spectra from a public database. And at the end, we will annotate our spectra and export this data again in a file format called MGF, so short for mascot generic file, which is commonly used to share MS2 spectra in mass spectrometry. Another information also here. So this I will walk you through this workshop, but there is also the possibility to run this whole workshop on yourself. So there's a Docker image, like usual with these workshops, that you can run locally in your computer or also in this orchestra setup system. And right, and with that, okay, I don't see any questions. I jump into the workshop itself. And before torturing you with R code, I want to give a very, very simple introduction to mass spectrometry and what type of data we are actually analyzing here. So really, just in a nutshell, what we have is mass spectrometry. We have an instrument that's able to measure charged molecules, so ions, and it will record the mass-to-charge ratio, or detect that the mass-to-charge ratio of an ion, and then measure the intensity, so the amount of this ion in the sample, and represent this as an intensity. So that's also the data that we get from a mass spec. It's also called a spectrum, where we have these 2 dimensions here, so mass-to-charge ratio on one axis and the intensity on the other axis. Now, for small molecule mass spectrometry or metabolomics, we usually don't have charged molecules. We have to generate ions first. This can be done, like shown here, by electrospray ionization. And another property of metabolomics, for example, is that we have many metabolites that have a very similar, also the same mass. So the mass spectrometer will not be able, or will have a very hard time to discriminate between them. So it can very well be that we are measuring different ions that have a very similar mass-to-charge ratio as a single peak. And for that reason, we are usually combining mass spectrometry with a second separation technique that uses some other property than the mass. And that's what's shown here on the left side. So we have here an example for liquid chromatography, or LC, where we can, for example, separate our molecules based on their polarity. And this setup is then called the LCMS setup. And with that, what we do is we measure now our sample for a certain time period. The mass spectrometer will, at discrete time points, record spectra. And the data that we get by this is then 3-dimensional. So we have the spectrum here. And the LC adds the 3rd dimension, which is the RT, or retention time. And what we get is, as I said, spectra for discrete time points. So we have multiple spectra for one sample. And that's also the way how we analyze the data. So we are analyzing the data here in what's called the MS1 data. We do quantification. We identify the so-called features that are then characterized by a mass-to-charge ratio and retention time. And the problem here is that we actually don't know what we are measuring, because for this setup, we don't have a label or a tag on our molecules. So we are left with this mass-to-charge ratio and retention times and need to find out what it could be. Or an additional possibility is to generate fragment spectra from the ions that give us a little bit information about the structure of these ions, of these features that we are measuring here. So what happens there is that the mass spec will then select for a certain retention time and mass to charge ratio, what's called then a precursor. So it will select ions, and it will throw these ions into a collision chamber where we have again an electrical field. These get accelerated. In the in this collision chamber, we have a gas, and these ions will then hit the gas atoms. And based on the energy that's applied, they will break down into fragments. And we can then measure these fragments as MS spectrum or MS2 spectrum or fragment spectrum. So we get here, for example, the masses and intensities, so the amount of these individual fragments of our precursor. And this fragment spectrum is now sort of a fingerprint of our molecule that's specific to the collision, so this fragmentation setup that we used here. And we could now start and match this, let's call it experimental MS2 spectrum against a reference library to really know what we measured there. And that's also the example that we will do in this workshop here. Okay, so that's very short introduction to mass spec. Now, short introduction to the Spectra package. So Spectra package is, let's call it, relatively new. So we added it, I think, 2 years or 3 years ago to Bioconductor. And basically what we wanted to do there is implement a very flexible and expandable infrastructure for mass spectrometry data, also learning a little bit from our expertise on MSnbase, XCMS, and MZR packages. So what we did now for the Spectra package is we completely separated all the code that's related to the data analysis, so what the user will usually use, and that's available for our object called Spectra. And we separated that from the code that's responsible to import the data, to export the data, and to represent the data, so how it's stored and where the data is stored. For that, we have the MS Backend interface, and we can have different implementations for that, like shown here. It can be plugged in, let's say, into the Spectra object to pivot the data. So that's the purpose of the backend, is only to provide the mass spec data to the Spectra object so that we can do something. And we will use here in this workshop different types of backends, and I want to explain you a little bit how they differ and why, and what the benefits are, what the different properties of these are. Right, so that's the introduction here. We said I'm going now to jump into the R code. Let me first start my Docker image here on my computer. So you could also start this on Orchestra here. So it's the list of all the workshops. And if you search for Spectra, you see that we have this workshop here. It's actually it's a new one, although we added it at the last conference, but this is the new version, so you can also launch it here. But what I will do is I will use the version from my local host. Let's log in here. And right, so here we are with the familiar look and feel of our R Studio. And inside of the vignettes folder here, we have the R Markdown document of this workshop that I'm opening now. I hope that, let me know if the size of the font is too small or too big. Let me also hide this thing here to make it bigger. Okay, looks like it's good. Then let me go down to where the workshop starts. So that's all the introduction, blah, blah. Oops, here we are. Okay, so the data that we are going to analyze in this workshop is provided by 2 MZML files that are within this package. And what we did here, so we generated these files in our lab. And what we did is basically buying pure standards for 8 molecules, so 8 metabolites. And these were added either to water or to a pool of human serum samples, and were then measured with the LCMS-MS setup that we use in our lab. And the MS2 data was generated for a collision energy of 20 electron volt. And so what we are doing here is, first of all, we load the Spectra library. Then we define these files. So these are the 2 files. They are also located within the package. So you see here the file names. And the next thing we are doing is essentially reading the data. So we want to generate a Spectra object. We give it the file names, and we also define with source, MS Backend, we define which backend we want to use, which backend can import data from this type of files. In this case, it's MZML files, so we use the MS backend MZR because that one can import data from MZML, MZXML, and CDF files. And what we have now is a, don't have anything. Oh yes, now my browser is stuck. Have here is a Spectra object. It tells me I have 1,500 Spectra in this data set. I am using MS backend MZR. And you see also that we have here already some information in addition to just the MZ and intensity values. So we have additional what we call Spectra variables available for each individual spectrum, like the MS level, the retention time, scan index. And if we go down here, it tells me that I have 33 more such variables. And now the Spectra object itself, so the data is, let's say, organized linearly. So each element is one spectrum. And what we can also think of is like this Spectra object being like a data frame with each row is one spectrum and each column would be one of the Spectra variables. And if we want to know what Spectra variables we have available, we can just use the Spectra variables call on our object and we get a list of all the variables that we have. So see, that's all this additional information that we have for each spectrum that is a little bit dependent also on the file type from which we import. So in this case, since it's from MZML, all this data, all this information is retrieved from that. And if we want now, for example, to extract a specific Spectra variable like R time, we can use either R time on our SPS all, which would return now a numeric vector of 1,500. So that's why I'm just appending here the head call to just show the first 6 of them. So, oh, sorry, now it's gone. And so this is one way how we can access data. And the second one, which is more, has a look and feel of a data frame, is that we can also say $R time. So like accessing the column of our data frame to extract Spectra variables. And you see that's exactly the same. And also here you see that's LCMS MS data. So we have a spectrum measured for every about 250 milliseconds here. So that's how we can access the data. Another thing that I just wanted to show here is, oops, sorry, that's always happening. I don't know why. Let's clean that up. So if we look now, for example, at the size in memory of this Spectra object, you might realize, well, that's pretty small, 0.5 megabyte for 1,500 Spectra. And the reason for that is that this MZR backend is not keeping all the data in memory. So only general spectrum data is kept in memory, and the actual master charge and intensity values, they are not in memory. So what happens is every time I use MZ or intensity call, they will be retrieved on the fly from the original data files. So you see here, we get, for example, all the MZ values for all the spectra. So each element in this list is a numeric vector with the MZ values for one specific spectrum. Good. So that's nice. So that's something that we had also in MSnbase with the on-disk mode and in mem mode. But the good thing, or the new thing, let's say, that we have here is that we can change the backend at any time. So we can, for example, say, oh, I'm not happy with having this MZR backend. So what I want to do is I want to change the backend. And that's what we're doing here. We just say, well, please use now, instead of the MZR backend, this MS backend data frame. And what happens is that we read now all the data from the files into memory. And you can see that because the size is now bigger of our object, it's still not big. And also, it's hard to realize, but also the access to the master charge and intensity values gets faster. So we have these different ways now to represent the data, either in memory or on disk. And obviously, the on-disk mode allows us also to analyze very large mass spectrometry data sets. And the last thing I wanted to show here in this data import part is subsetting. So subsetting is also very convenient, let's say. So we can subset a Spectra object in any, with a square bracket operator like any other object in R. And I can give it also arbitrary indices. So they will be returned in whatever order that I provide here. So that's one way to subset a spectral object. But sometimes better way to do that is to use one of these many filter functions that we have that allow very specific subsetting operation that are commonly used for mass spectrometry data, and also they are optimized, so they are sometimes really fast and more efficient than the square operator. And in our example, what we do here is, so we want to subset we want to essentially extract from this 1,500 spectra that we have. We want to identify those that were generated from a precursor ion that matches this mass-to-charge ratio. And this is the mass-to-charge ratio that we would get for a certain ion of a metabolite called cysteine. And in fact, it's a M+H ion, so it's the metabolite plus a hydrogen atom, so basically a protonated ion, and this would be the mass-to-charge ratio. So what we do here is we use the filter precursor MZ function, give it our the big spectral object with the MZ. We can provide a range of mass-to-charge ratios that we want to filter on. So this call essentially takes our mass-to-charge ratio of interest and adds a small error to that, so in ppm 10, so it's 10 parts per million difference that we accept for our filter. Sorry. And with that, what we get is now a spectra object that has only 6 spectra left in it. And here we see, well, we see these 6 spectra. And that's the small subset that we will go on to analyze now in the next section. So this was more about data import and just explaining a little bit these 2 different concepts of keeping the data in memory or on disk. Now we come to data processing and manipulation. So that's also something we want to do. And before actually manipulating the data, it's also sometimes good to just visualize the data. So with the plot spectra call, we can plot a spectrum. Maybe it's a little bit too, just make the line width thicker. And so that's the first spectrum from our 6 selected spectra. And you see here, as expected, we have intensity values, mass-to-charge ratio, and we can see some very nice peaks here, so fragments with a quite high intensity. But what we also see is very low intensity peaks around here, and that's something that I don't really want to have here. So for matching and annotation purposes, this doesn't really help. That's just noise that I would get rid of. And to do that, we have a function which is called filter intensity. And this filter intensity function would allow me now to select a threshold below which all the peaks, so intensity threshold, would be removed. But I would like to do a little bit something more sophisticated. So what I would like to do is to remove all peaks that have an intensity which is lower than 5% of this highest peak in a spectrum. And the good thing is I can also do that with the filter intensity function. So the first thing I do here is simply define a function called lowInt that takes intensity values, so numeric vectors input, and then compares these intensity values, and the output should be a logical vector of the same length than with this input vector here that tells the function whether to keep or remove this specific peak. So in this case, we just compare now the intensities and check if these intensities are higher than 0.05 times the maximum intensity. And when we apply this now to our spectral object, we can call filter intensity by passing this function as an intensity parameter. And if we plot now the data, again, where is my plot call? We see that we have now removed all these low intensity peaks. So I'm quite happy with this result here. But there is also another thing that I observe, which is I have quite high intensity, so 120,000 and so. And MS2 data is usually not really quantitative. So I prefer to have this data sort as percentages. So I want to have the highest intensity set to a value of 100 and all the other intensities here, all the other peaks to be relative to that. So that makes it also easier for the spectral comparison because I'm only looking for a pattern and not for absolute values here. Now, the problem we have, or it's not a problem, but we don't have a dedicated normalize or scale function in spectra. But what we have is actually something more powerful. So we have a function which is called add processing. And with add processing, I can apply any data manipulation function to the data. So I can define my own function that takes a peak matrix from one spectrum as input. So the peak matrix is essentially a 2-column numerical matrix, with one column being, so first column, the master charge ratios, second column, the intensities. I can then do in this function anything that I want with this peak matrix, and at the end I have to return again this 2-column or a 2-column matrix again. So in order to normalize now our spectra, I define here a simple function, call it normInt, where I first calculate the maximum intensity. So this X is now the peak matrix. And then I replace the intensity column in this peak matrix by dividing the intensities by the maximum intensity times 100. So I have them relative intensities. And then I return this again back. And I add now this processing. So we have now a spectra, add processing. And if we plot this data, again, we see we have now intensities which are going from 0 to 100. So this works very nicely. And right, so also if I look now at my Spectra object, it tells me always that what different, maybe I explain this later, what different operations I have done. So we always have a log of all the processings that have been applied to a Spectra object within the object itself. Okay, then next very short section. So as I said, the goal would be to match now our, what we call here the experimental fragment spectra against a reference, and comparing spectra, how can we do that? And obviously we have a function for that, which is called compare spectra. So the name tells already what we are doing here. And this compare spectra, simply in this case, since we are passing just the single spectrum object, it calculates a pairwise similarity between each of our 6 spectra. With ppm 20, I can specify the maximum allowed difference in master charge ratio for peaks to be considered matching. Because obviously in mass spec, it's not like if you repeatedly measure the same ion that you always get the same master charge ratio, but there is a small error in the instrument, and this ppm allows us to specify that. And also what we do with that, so by default, we're using here the normalized dot product as a similarity measure, but there's a large number of other similarity calculations available in Spectra. So just have a look at the help page to get more information about that. In our case, what we have is result is a similarity matrix. So 6 columns, 6 rows, each element compared against each. The diagonal is comparison with itself. And what we see here quite nicely is that the similarities are very high. We can also just, it's maybe easier to see, we can also visualize this in a heat map. So all the similarities are at least above 0.8, which is actually a good thing, because this means that these 6 fragment spectra most likely represent fragmentation of exactly the same metabolite, which is not given, because if we look, for example, at the retention time of our 6 spectra here, we see that they are measured in 10-second differences. So it could well be that, for example, here after 231 seconds, we would by chance measure a metabolite that by chance has exactly the same master charge ratio than the one that we had here at 220 seconds, but then we would have a different fragmentation profile there. So we could identify that with this. In our case, we are all fine, and we can now proceed with the main task of this whole workshop, which is I want to compare now my experimental spectra against a reference. And the reference, usually what people have is they have their in-house libraries, like also what we generated here with this experiment. So fragment spectra for certain known compounds generated with our LCMS setup, so that we can really be sure that we know what we measured there. But it's now, there are also additional resources now becoming available. that provide you with publicly available fragment libraries, so Spectra, that are growing over time, obviously. So it's always when people publish MS2 data, they start adding these to these databases. And I've just listed here some of them. So one of the main one is MassBank. We have MONA, Human Metabolome Database, GMPS, and many more. So that's very nice. It's a very good thing, so that we have also here public spectral libraries against which we can compare our experimental spectra. The problem I have with that is that each of these resources defines their own data type and their own way to store and make the data accessible. So MassBank saves, stores data, or provides you the data in their typical MassBank specific text file format. MONA uses JSON. HMDB has their own XML file format. GMPS uses MGF files. So there's no common, let's say, interface. So it's not easy for the, let's say, end user to integrate such resources into the workflow. And that's actually where we then started to implementing now MS Backend instances for these databases, so that we can integrate this data easier into a Spectra-based workflow. So far we have implemented a MassBank and HMDB backend, but the others, maybe if I find time, we might also implement them. And so these are available in separate packages. So to use the MassBank, what we are doing here, we use a package called MS Backend MassBank. And this one provides two backends. So one is called MS Backend MassBank. This allows to import and export mass spectrometry data in the file format from MassBank. And we have also this backend here that we will use in this workshop, which is called MS Backend MassBank SQL. And that's actually quite nice, because this one allows me to directly connect to a MySQL database that has all the MassBank database in it. So it's more convenient than downloading everything beforehand. And so that's what we are going to do in this workshop here. So first of all, we load the library MariaDB to connect to a MySQL database. Then we load this package here, and we connect to the database. So this is the cool thing about this Docker image. So I've bundled a MySQL server into this Docker image that has an installation of the MassBank release 2021-03. So that's the most recent one. And we can connect to this database. So we really have now a database connection here. And we could also call SQL calls on that. Like here, listing all the tables that are in the database. But that's not really convenient to the end user. So trying to find out where, in which table, which information is stored. So what we do here is now, like before, we create a Spectra object representing MassBank. So in this case, I've called Spectra, give it the connection to the database, and define to use this MS Backend SQL. And what I get here is now a Spectra object that has the 86,000 spectra from MassBank. It uses this backend here. And I have the spectra variables, MS level, precursor, master charge ratio, and 42 additional ones. So this really is all the annotation that's available in MassBank is also available now in our Spectra object. So I find this actually quite cool and useful. And yeah, so things that we can do with that is obviously we can call or extract like on our mbank 1 to 10. We could, for example, extract the precursor MZ, master charge ratio. So we have, for some of them, we have some, for others we don't. We could also call MZ on our mbank on the first 10 spectra there. And you see, we get all this information like we had before for the experimental spectrum. The only difference that we have now is that in this case, everything is always retrieved on the fly from the database. So no data is stored here in memory. Everything comes from the database. And so every call that we make on MZ will query the database to extract the data. Good. So another thing that we might realize here is that I have here values that are higher than 100. So again, we are left with absolute intensities, and I would like to have everything now the same scale. So also my data, I did some data processing, And the obvious thing to do is now, well, then let's just apply the same processing to this mBank. So I add this add processing to the mBank, this norm int function again to normalize the intensities. And if we now extract the intensities again, we see they are now really scaled, but add processing norm int. They should now be normalized. Let me see. 10. Okay, something went wrong here. I don't know why. Let me plot that. Oh, it worked. I don't know why the query is showing something different. So we have now everything scaled to values between 0 and 100. And while this looks obvious, it's in the end, it's not so clear because we did now data manipulation on a object that where we don't actually can change the data because the data is in a MySQL database and people of MassBank definitely don't want you to change their master charge or intensity values in their database. So this is something that we also added to the Spectra package. Basically, it's called the delayed operations, or what we call it, the lazy evaluation queue. So we have here a processing queue in which we store the processing steps. So any data manipulation that we do is not applied immediately to the data, but cached inside the object and only applied to the data whenever we extract the data. Okay, so now I have now my processed MassBank data. I could now start comparing my 6 experimental spectra against all the 80,000 MassBank spectra, but it's computationally not very efficient and also doesn't really make sense if there is not a hint that this might be the same fragmentation. So what I'm doing here is, again, using the same command than above to filter this case MassBank for all the spectra that were generated for a precursor mass-to-charge ratio that matches the ion that I'm interested in. With that, what we get is, again, a spectra object, and we have 12 matching spectra in MassBank that match our precursor. We see it's all the same precursor mass-to-charge ratio. And with that, we can now go ahead and compare each of these 12 spectra against our 6 experimental spectra, and we do this like before. So we have compare spectra, we give it our MassBank spectra object, our experimental spectra, and as a result, we get now again a similarity matrix that has, in this case, 12 columns, 12 rows, 6 columns. So each MassBank spectrum compared against each of my experimental spectra. And you see we have some quite low similarities here, but also here some that are very high, like 90% similarity. So we definitely found a matching spectrum in MassBank. And we now want to identify the best match here in this matrix. So we just look which cell here has the maximum similarity. And we do this down here. With r.index, we return this the index as a row column index. So we have here the 5th row, so the 5th column from MassBank matches best with the 5th of our experimental spectra. And right, so we are now quite happy, and we would now like to annotate, or actually, yeah, let's also plot it before. For time reasons, I will not explain how exactly we plot. So this is just, again, oops, sorry. Let's plot it again. It's just a graphical visualization of this similarity. So we have in the upper panel the MassBank spectrum, in the lower panel our experimental spectrum flipped, and the matching peaks based on their mass-to-charge ratio are highlighted in blue, and we see that almost all peaks match here, and also their intensities match. So we can be really sure that this fragment spectrum that we measured matches the one from MassBank. And so we most likely have the same, or we have the same compound measured here. And right, so if we want to annotate now our own spectra, we can have first a look what annotations is available in the MassBank. So we see a lot of information, like also the publication in which this spectrum was found. And here the last one is actually the compound name. So this tells me what was the metabolite that was measured. So this is the information I want to add now to my Spectra object. So basically I want to annotate it. So first what I do here is subsetting this whole mass bank for the best match. And then we extract the name of this. So in fact, it's cysteine, what we expected. And now I want to add this information to my Spectra object. That's really simple with Spectra objects. Because like I said before, like with a data frame, if you want to add an additional column, you just use $ and the name of the new column. So we can do the same with the Spectra objects. We just say SBS $ and the name of the new Spectra variable we want to add. So we add here like name with formula, the chemical formula, adduct and collision energy. And we have then this additional variable stored inside the Spectra object. So we have now a beautiful annotated Spectra object that we would now like to export because we want to share it with collaborators or just store it somewhere. And as I said in the beginning, we would like to export this in MGF format because that's one of the commonly used formats for exchange of MS2 data. And now Spectra itself does not have support for MGF files. We cannot read and export it, but Laurent actually implemented this nice package, MS Backend MGF, with the same named backend for Spectra that allows us to import and export files in MGF format. So to export this now, we simply call export on our Spectra object. With the backend, we specify the backend that should be used for the export, and we give it also a file name. So by specifying here MS Backend MGF, we actually tell or we define to export the data as an MGF file. So this determines the file type in which we want to export the data. If we would have used MS Backend Mass Bank here instead, then we would have exported this as a text file in mass bank format. And if we would have used, for whatever reason, MS Backend MZR, we would have exported this data as an MZML file. So that's how we can export the data. So we are quite happy with that. I will jump that section. That's just repeating the same thing that we did for mass bank using the human metabolome database. And then we'll go to the very end, to the summary, actually the short summary. So these are the two main, let's say, properties of the Spectra package. So the first thing that we did is really separating all the functionality for data analysis from the functionality that's responsible for data storage. And this really allows us to add very easily additional file format, import, exports, or data representations to a Spectra object because it can be provided by other packages. And then we have also this caching mechanism so that we can really apply data manipulation, any data manipulation to data, even if the data is not locally on your computer, but is somewhere remote in a database or on a remote file system where you don't have the rights and the access to change the data. And at the very, very, very end, just mentioning some further developments. So there's some additional packages also built upon Spectra. So it's metabo annotation that Andrea and Michael are developing that provides actually an easier workflow. So easy functions that simplify the workflow that we used here. We have compound DB that allows me to create small compound annotation databases, including Spectra object. We have MS experiment to represent whole experiments, meaning the raw data along with sample annotations for the files and identification data, everything in one object. Thomas is currently developing a nice package for quality assessment of mass spectrometry data. And we have additional backends that are being implemented or are available for different other file types. And with that, I am actually done and would hand over to questions. Actually, I maybe stop sharing here if I manage to do that. Let me see. Oh my God. So I see already some questions here. Thanks. Post-translational modifications. Laurent. I mean, I'm not working in proteomics, so I'm not sure that Spectra can do anything with post-translational modifications.
So in terms of PTMs, these would be, I mean, they wouldn't be identified. You wouldn't get your identifications of peptides and PTMs in Spectra. You would use a third-party application for that that would generate these identifications, typically in MZID format. And that MZID format can be imported into R as a data frame-like structure. And that one will contain your peptide identifications, including the modifications. And that is something that you can then use and kind of attach or join to your Spectra object, so that you can, on one hand, have the raw mass spectrometry data as has been demonstrated by Johannes, and each of these spectra would then have their respective proteomics associations, annotations, which are peptide sequences if you have a PSM, including modifications if they were present in that peptide.
Okay. Man, again, proteins. Okay, how's right? I mean, go ahead. Yeah.
Yes. So again, that would be something that would be handled outside of Spectra. I think that's typically something that you would, if you know about your modifications, that they induce changes at the protein level, when you run your identifications, you could indeed use multiple protein sequences in your protein FASTA file per protein. And so some modifications that include the modified, so some sequences that account for the modifications you're interested in. And that will allow you, or hopefully, to identify the modified peptide, right? Or the non-modified peptide, because you will have the two peptide sequences in your FASTA file. And so, as I mentioned before, these peptide identifications, with or without modifications, could then be attached to your raw data that is stored in a Spectra object. And the package that handles these identification data is called PSM. It's not on Bioconductor, but you'll find it on the for mass spectrometry GitHub organization. The name will have to be changed because the PSM name already exists in a CRAN package that has been orphaned or removed from CRAN for a couple of years now, but I know it's already taken. But so again, this is something that you would handle way before analyzing your data when you plan your experiment. I hope this answers the question.
No, actually, I like the question, so it's not out of scope. It's just one thing that I should also mention. So for now, this Spectra package is more like infrastructure that should give you the tools to do all these things by yourself. So you can remove certain peaks, add peaks, you can play around with the mass spectrometry data, which I think in that way it was not really available before. So to have this power to, just add or remove individual peaks or change their intensities and master charge ratios are in a very flexible way. And I hope that we could really do this now with the Spectra.
And I think the presentation, which was more metabolomics oriented, and some of the questions that are proteomics related, really show that this collaborative development, kind of this collaboration worked because it's of interest to different communities, although they use the same technology. But to that extent, I think it's a nice deliverable.
Right, and also, I mean, if you have any more questions or just want to have a chat, I think every now and then we will be around on a table in the launch, which is called the Biomedicine mass spectrometry. So in case you want to have a chat later. I think we have 13 seconds left. So any urgent questions, or we release you to grab a coffee before the next workshops. So thanks a lot for your attention, and see you around. Thanks.
Thank you very much, Johannes.